Hello everyone, my name is Cecilia Barbieri. I'm doctoral student at the Department of Chemistry and Biochemistry of the National University of Mar del Plata. On this occasion, I'll show you a work called Rational Drug Design applied to the search for candidate to reverse multidrug resistance mediated by P-Glycoprotein, hereafter a PGP. Multidrug resistance is a major obstacle to the success of chemotherapy. One of the main mechanisms involved in this phenomenon is the overexpression of PGP, a transmembrane ATP binding cassette protein that pumps a wide variety of anti-cancer drugs out of the cell. From the bioguided assay of the Ligaria cuneifolia plant made in our group, the compound betulin emerged as a promising inhibitor. Molecular modeling studies revealed that betulin binds to a key region of the transmembrane domain with a binding mode that overlaps a major doxorubicin site and emulates the same contacts as the reference compound, tariquidar. In another work studying the structural activity relationship, certain structural determinants which confer inhibitor activity were characterized. By combining those previous studies, a series of novel betulin derivatives were designed and synthesized to find the best candidates. Docking and molecular dynamics assays were performed to obtain information on how the ligand interacts with PGP. We were able to divide the compounds into two groups, those located at the same site as doxorubicin in green and those located at another site in magenta. In terms of binding energy, the green-colored species were found to effectively inhibit the efflux of rhodamine 123 and doxorubicin, being comparable to or better than verapamil, a no inhibitor that is not suitable for clinical trials due to its side effects. The magenta-colored compound were inactive regardless of their binding energy. The green-colored compounds showed persistent contact at the residues of both homologous hubs, both connecting the transmembrane domain with the nucleotide binding domain. The power of this compound and the lack of any activity for those which went to the other side confirmed the relevance of this site, which was proposed years ago by our group, before appearance of the experimental structure of the complexes PGP taxol and PGP pancreatine. The ability of steroid moiety to fit in a hydrophobic cavity, the flexibility of a linker to the substituent, the number of methoxy groups, and an optimal length of the molecule in its extended conformation were found to be the keys for improving the activity of the betulin scaffold. In conclusion, the modeling was worth for obtaining powerful lead candidates with lower or null toxicity and to understand the main factor which confer the MDR inhibitory activity. Thank you so much. See you.